Okay, so this is chapter 21, objective 4, part 2. So hopefully you've already seen objective 4, part 1, because in this section we're just going to talk about the T lymphocytes. So just reminding you all, this was a slide previously in one of the previous objectives, maybe objective 1, I don't remember, but it was explaining the difference between humoral immunity versus cell-mediated immunity. So remember, cell-mediated immunity is dealing with pathogens that are located inside of cells, where humoral immunity is dealing with pathogens that are located on the outside of cells. So B cells are primarily dealing with outside, where T cells are dealing with what's on the inside. Okay, so when we're talking about cell-mediated immunity, we're talking about T cells. So just to kind of remind you all about the cell-mediated response, may not have used those phrases in the previous video, but keep in mind you've got your antigen-presenting cell, such as your macrophage. Remember, it could have also been a dendritic cell, is going to engulf the pathogen. It's going to be presented on the surface of that cell by a major histocompatibility complex. T cells are then going to be responding to that antigen on the surface of that antigen presenting cell. And T cells are going to secrete cytokines. That's going to cause more T cells to reproduce. And then your infected cell will then die. Okay, so the primary cell involved in adaptive immune response are your T and B cells, okay, T and B lymphocytes. T cells directly control the immune response and also control the B cell response. So T cells control B cells. Each T cell is only going to produce one kind of receptor, which makes it specific to a specific antigen. And there are several parts to that receptor. So you've got right here in this image, you've got a cancer cell. Remember, any cell in the body can present a major histocompatibility complex 1. Okay, all cells that are nucleated can do that. So this cancer cell could be a skin cell, for example. Your cytotoxic T cell, which is a type of T cell, cytotoxic T cell is going to respond to that immune cell, which is specifically cytotoxic T cell, and it's responding to that major histocompatibility complex 1. So based on the image, we can infer this is a cytotoxic immune cell. That receptor has got alpha and beta parts, which is what's going to specifically bind with that antigen. That's your variable region, and then it's got a constant region. Okay, antigen presentation. You guys already know this. We've already talked about this. So T cells only recognize antigens when they are on the antigen presenting cell. So this antigen could be floating by in the bloodstream, and the T cell is just going to let it go. So it's only going to be aware if it's on the antigen presenting cell. If it's on a pathogen floating by, nothing. It's got to be on the antigen presenting cell for the T cell to recognize it. Antigens are internalized by the antigen presenting cells. We talked about how that was happening with the phagocytosis of the dendritic cells and the macrophages. The antigen presenting, I'm changing my thing here. The antigen presenting process takes place when the antigen is broken into smaller pieces, which we've talked about. Those fragments are then taken to the surface and presented with a major histocompatibility complex. It could be a 1 or it could be a 2, right? If it's a class 2, we know that it is a professional cell. All cells that have a nucleus can present an MHC class 1. The MHC class major histocompatibility complex in conjunction with the antigen is placed on the surface, and that is antigen presentation. So keep in mind the major histocompatibility complex 2 is going to communicate with T helper cells. T helper cells, also known as CD4 cells. The major histocompatibility complex 1 is going to communicate with the cytotoxic T cells or the killer cells, also known as CD8. Okay. Now, what is the difference between MHC class 1 and MHC class 2? There's a super long video for MHC class 1, and there's another super long video for MHC class 2s. You all are welcome to watch those if you want. They are very lengthy and have probably more information than you need, but it's something else if you want access to something else to watch. Major histocompatibility 1 is intracellular antigens. So this is when a virus or some particle goes inside the cell. And remember, this is dealing with every cell in the body because every cell in the body can exhibit this MHC class 1 receptor. So this is when basically your cell of your body gets infected with some type of pathogen. This stimulates the cytotoxic T cells. And since viruses can affect any tissue, all tissues have to be able to express this MHC1 or no T cell response is going to occur because, again, T cells only respond to antigens presented on the surface by MHC1s, okay, or 2s. So MHC2s are extracellular antigens that are ingested by antigen presenting cells, the professionals, okay? They're going to undergo phagocytosis, bring these particles into the cell, and then express those antigens on the surface. So these are extracellular antigens that have been brought in. These are viruses that have infected the cell and now you have a sick somatic cell. The somatic cell is sick because something went into it. Okay, T cell development. We talked about T cell development in one of the previous lecture videos. Remember T cells, well, and also in chapter 17 when we talked about thymosin. So thymocytes, that's a fancy word for an immature T cell. Okay, so instead of calling it a T lymphocyte, it's a thymocyte. It's an immature T cell. These leave the bone marrow. Remember, all white blood cells, red blood cells, platelets, they are made by stem cells in the bone marrow, the red bone marrow. It's going to leave the bone marrow, these thymocytes, and they're going to go to the thymus gland. Now, cells in the thymus gland present MHC proteins to the untrained thymocytes. Remember, T cells recognize MHCs. You have to know that your T cell can recognize the MHC proteins that are expressed on the surface. So the thymus does that. It throws out MHC proteins to see if these thymocytes can recognize them or not. So the thymocytes that can correctly identify MHCs are separated from the ones that cannot. So if the thymocyte is unable to recognize an MHC protein, it's destroyed. Apoptosis, meaning it's programmed cell death, the cell self-destructs. This is called positive selection because we're only keeping the thymocytes that actually can bind to the MHCs, that can recognize it. So now that the thymocytes can recognize the MHCs, they are awarded a prize by either getting a CD4 and a CD8 receptor. So they're going to get these receptors now. They're the little prizes. Thymocytes go deeper into the thymus, where antigen-presenting cells present self-antigens to the thymocytes. The thymocytes that bind with the self-antigens undergo apoptosis. Why would you kill a thymocyte if it bound with a self-antigen? Remember, self-antigens are your antigens. We don't want T cells to destroy self-antigens. We want them to know, oh, okay, this is part of self. I don't destroy it. So any thymocyte that has the ability to bind to a self-antigen gets destroyed. Those that don't bind, they move on to the next section. This is called negative selection because those that don't bind get to move ahead and advance, right? The other one's apoptosis. So cell signals cause the self-tolerant thymocytes to express either CD4 or CD8. Those that express CD4 become helper T cells and will bind to MHC2 receptors. Those that express the CD8 receptors are called cytotoxic T cells, and they will bind to MHC1 receptors. Believe it or not, of the thymocytes that enter the thymus, only 2%, this is amazing, only 2% actually leave the thymus gland as mature T cells. 98% of the thymocytes that enter the thymus gland get destroyed by the thymus gland. So not good, not a good turnover ratio here. So your T cell can either be classified as a T helper cell or a cytotoxic T cell. So this cytotoxic T cell, also known as a T killer cell, okay? So the T helper cell expresses the CD4 receptor, the cytotoxic T cell or the T killer cell expresses the CD8 receptor. So again, we've got the helper T cell, which is expressing the CD4, and it's going to bind to the MHC2 on the antigen presenting cells. And again, those are the professionals. The cytotoxic T cell expresses the CD8 receptor and it binds to the MHC1. And again, on an antigen presenting cell, which again, this could be any cell in the body can express this MHC1. So helper T cells, there's actually two types. There's Th1s and Th2s. And then your cytotoxic T cell just has these cytotoxins that are produced that are within the cell, where the helper cell undergoes exocytosis with those chemicals. Chemicals go outside of the cell, and they are called cytokines. So we have our antigen presenting cell here with our MHC2, which means, oh, that's a professional, probably a macrophage or something. We have the helper T cell. We know it's a helper T cell because it's got that CD4 receptor on it. And once activated, right, once it binds to that antigen presenting cell, and receives some chemical stimulation there, it will become active. And the helper T cell does some amazing things. It virtually stimulates and activates every other immune system cell. So your T cell goes out and sends signals so all of your immune cells know, hey, we got something going on in the body. We've got to all get ready to go. So we also make some memory T cells, right? Because it's recognizing this antigen right here that's on that MHC2 presenter. So we're going to make some memory T cells so we don't have to go through all this next time, and we're aware. So we're going to put those in the background. But it's also going to secrete this cytokine, and there's all different kinds of cytokines. And depending on what cytokines are produced from these T cells, and they have the ability to produce all these different kinds of cytokines, is going to bring in different troops. So certain cytokines or interleukins that are produced are going to come bring in the neutrophils and macrophages for help. Other cytokines that are going to be secreted are going to bring in the natural killer cells, the cytotoxic T cells. And then other cytokines are going to let the B cells know, hey, it's time for you to come in. So remember, the T helper cell is going to bring all these other immune cells to the rescue, because notice it undergoes, these chemicals are released through exocytosis from that helper T cell. And those are signaling molecules to let everybody else know it's time to come on in and join the party here. We got to get some stuff done. So again, T helper cells express this CD4 protein on the surface. This plays a role in activation. It's going to communicate with the major histocompatibility complex 2 on the antigen presenting cell. So that way it can recognize foreign substances. Now, there are actually two classifications of T helper cells. You got T helper 1 and T helper 2. And based on what they secrete is going to depend on whether they're a T helper 1 or a T helper 2 cell. Okay. I am not going to worry about specifics on this. So you do need to know, you can identify the T helper 1 versus the T helper 2 based on the chemicals they secrete. And all of these terms right here, which looks like gibberish, that is telling you what the specific cytokines are that are being secreted. IL stands for interleukin, IFN stands for interferon. You got tumor necrosis factor. We've got over here with the T helper 2 cells, just a bunch of interleukins. So there's more than one kind of interleukin. We're not getting into all of that. I do want you to notice, though, the T helper 1 cells, what did they call in for activation? Your professional antigen presenting cells. So they get the cytotoxic T cells, the B cells, and the macrophages involved, whereas the T helper cells, they're involved in primarily what we're going to talk about here. These are non-specific, but the B cells are specific. B cell activation. They're going to activate B cells. Okay. So two types of helper T cells based on the cytokines they secrete. T helper 1 cells, they work on that cell-mediated response by bringing in cytotoxic T cells and macrophages. Remember, cell-mediated is targeting pathogens that are inside the cell. Remember, these were engulfed by the cell through phagocytosis and expressed on the surface by MHC2 receptors. Now, the helper T cells class 2 stimulate B cells. Those are part of the humoral response. That's dealing with infection outside the body, or I'm sorry, outside the cells. And this is going to stimulate B cells to make antibodies. So these T helper cells, man, they do a lot. This is the part down here that I'm not going to have you specifically know. I'm just putting that out there kind of for your information. But the helper cells secrete all different kinds of cytokines, interferon, tumor necrosis factor, interleukins, whereas the T helper cytokines pretty much are just interleukins, different kinds of interleukins. Okay, so the overall thing here, we've got a helper T cell. What's it ultimately going to do? It's going to bring in more helper T cells, and it's going to secrete cytokines. Okay, cytokines are going to activate more T cells and cause T cells to multiply, and then T helper cells also activate B cells. So you've got your little guy right here, which is your T helper 1 cell, and it's letting everybody know, hey, I found the antigen, or I'm sorry, this is your dendritic cell, your antigen presenting cell, and your T helper cell over here is recognizing it, and it's saying, hey, T helper 1 cell, get over here. I found an antigen. So the T helper cell looks at the antigen and says, hey, I've got to tell everybody what's going on. It does so by secreting different cytokines such as interferon, tumor necrosis factor, leukotrienes, chemokines, and these monocytes here, macrophages, are going to be alerted. That's just one type. It'll alert a whole bunch of people. Now, the other part of that is, guess what the HIV virus infects? Okay, y'all probably know it's T cells, but we got a couple of different kinds of T cells here. So which ones? Specifically the T helper cells. And remember, the T helper cells are the ones that alert everybody that there's a problem. So if you knock out your T helper cells, you're knocking out a big part of communication with the rest of the immune system. So HIV. So I've got a picture here of what that HIV virus looks like, and then a microscopic image. It's probably more of a closer image than just a microscope. So HIV infects the T helper cells, also known as CD4 cells. Why CD4 cells? Because they express CD4 on the surface. Keep in mind, T helper cells help fight infection and disease, right? HIV is a virus, which means it cannot replicate on its own. It attaches itself and it infects the T helper cell. It attaches to the T helper cell and goes into the T helper cell. And it takes control of the DNA, causing the T helper cell DNA to start making copies of the virus. So it turns into a viral replicating factor. It's no longer doing the job of the body. It is replicating HIV viruses. Those HIV viruses are going to multiply, go throughout the body. What this does is it weakens the body's natural defenses over time because it's severely damaging the immune system. You just infected the T helper cell. It's now replicating HIV viruses instead of doing its job. Okay. When HIV enters its genetic information into the helper cell, it starts to make copies of itself. So the HIV virus is replicating itself. When this happens, the helper cell dies. Which disrupts the immune response. Because again, that T helper cell was what was signaling all the other immune cells it was time to do something, right? So it was the signaling molecule for, everybody else come on board and start doing stuff here. That means the white blood cells, all the other cells in the immune system are not receiving very much information. And then that means that people with full-blown AIDS, meaning most of their T cells, they got a very low T cell count. You're going to be prone to more secondary infections. And you're probably going to die of a secondary infection, such as something your body would normally be able to fight off. That's why folks with pneumonia end up dying if they've got AIDS, because they don't have an immune response anymore, because the helper cells aren't there to tell the body what to do. Okay, this just kind of shows you a picture of what's going on. I'll let you take a look at this on your own. Just kind of shows you how it injects itself into the helper T cell, and then what happens. So it's just kind of the steps. I'm not going to ask you specifically. It's just kind of more information, but I'm not going to ask you any questions specifically off of this slide, but it just gives you a visual of what's going on. Pretty cool video. So you can see what's happening. Now, the interesting thing is, with HIV, it is it's an RNA-based virus rather than a DNA-based virus. So it's interesting that RNA is being injected into the host cell rather than DNA. So what your body has to do, what your cells are doing, is they're taking that RNA that's injected by the HIV virus and turning it into DNA, and then injecting it into implanting it into your DNA. So they're taking your DNA, splitting it, and sticking this DNA strand in there. So your body has to actually take the RNA molecule that is injected and construct DNA from that. Now, if you guys remember, that doesn't happen. What the process happens is DNA forms RNA. You don't normally get RNA forming DNA. That's not the way things go. So it's interesting that HIV takes RNA and forces your body to make DNA. It's reverse transcriptase. Okay, so then we talked about the T helper cells. Now we're going to talk about the cytotoxic T cells, and they're also known as killer cells. They express that CD8 receptor on there. So how do they work? Well, the cytotoxic T cell, again, a specific type of T lymphocyte, which is a type of white blood cell. This specifically kills cancer cells and cells that are infected by viruses or damaged in other ways. So this is targeting body cells that have been infected, okay? Infected body cells. Not your professional antigen presenting cells, which are your T helper cells. This is your non-professional antigen presenting cells, which have been infected by some type of virus or pathogen. The cytotoxic T cells express the CD8 receptor. They bind to MHC1 receptors. Remember, those are going to be non-professional antigen presenting cells. Any cell that has a nucleus can do that. Now, how does the cytotoxic T cell kill the infected cell? It induces apoptosis. It induces the cell to kill itself. It uses the same mechanism as those natural killer cells. So when exposed to an infected or dysfunctional body cell, somatic cell, the cytotoxic T cell releases cytotoxins, perforin, granzymes, which induce apoptosis, programmed cell death. And so your infected cell will die, not undergoing mitosis and distributing that, or replicating that issue. That was Objective 4, Part 2. See if I can get this done a little bit quicker than I did the last time.